one cat coming in. Right. Right, let's see what we're going to do this morning. That's me washing done. Oh, I've got to go and have a shave. I've got 5 minutes. Do. Bother with that one. Right, let's go and have a quick shave. I got how long?
4 minutes.
4 minutes. Ooh, we're a bit tight. Okay.
Well, I did it from the clock.
Yeah, that's all right. How long?
Apparently 30 seconds.
Alright, I'll be ready. Oh, you are a bloody nuisance, cat. Go on then. Right, you should be able to start on time. If I can get my pocket in. Perhaps not. Right, yep. Right, good morning. We're going to have a go at looking at a bit more A-level biology topic by topic. I'm going to start having a look at things like recombinant DNA technology, playing around with the PCR machine. And this is really quite relevant at the moment with the COVID-19 going on and how a lot of the testing needs to be done. So let's dip in and have a look at what is involved in this recombinant DNA technology. So let's have a look. Basically, what we need to do is to try and somehow isolate the bit of DNA that we want to try and have a look at. And we need to somehow then clean this DNA up so that then we can basically somehow copy it so that we've actually got enough of it that we can actually do something with it, whether it is to just identify or whether it might be to insert it into some sort of other organism. So we need to sort of understand what goes on, and for most of the work we need to understand a special enzyme, and this enzyme is basically enzymes like DNA polymerase. DNA polymerase is basically this sort of gene creation piece of protein, and what it can do, it can take a piece of DNA and increase it. What we can do is we can play around with this to make it work effectively. Let's have a look and see what goes on. It's all right. I was just looking for where I put my pens and I've lost them. Right. I had some pretty coloured pens around, which I've put around somewhere really carefully. Let's have our stage divided into basically 3 stages. And if we want to think about these, then we've got basically our step 1, 2 and 3. Right, now these 3 stages are, no, I don't want those. I want, it's alright, I've got some people trying to help me here, trying to find where I put my pens, and they're not there now. I had them out earlier. Don't worry, we'll do it. Now, let's take some sort of DNA fragment, and this DNA fragment we're going to split this DNA fragment into 2 strands. And magically my coloured pens have appeared. And we can draw this in glorious Technicolor. So I've got my base strands. Let's just have a couple of colours in here that might make this a little bit easier to sort of see what's going on. There we are. So we've got a few colours. Now we heat this up to 95 centigrade, 95 Celsius. And at this point what happens is that the DNA strands, they basically separate. Now once we've done that, we can cool the results down to 55 centigrade, 55 Celsius, and we've got then our piece of DNA, in fact 2 pieces of DNA here, and what we're going to do with these is we're going to try and add some sort of primer to them. I'll make that the right colour. There we go. We've got just some colours in there. I don't think they're really significant of any particular bases. And we take in a DNA primer. And to fit on here, then I'm going to want a piece that's going to be like that and like that. And this DNA primer will fit onto my piece of DNA. My strand of DNA is probably going to be a little bit longer than this. And a DNA primer will also fit on the other end. The colours here aren't significant, they're just the opposite base pairs. And this now happens at 55 Celsius. Step 3 is perhaps the most important of these phases, and what happens now is we have in here lots of different nucleotides, and they combine with this piece of DNA that we've already made. Let's just be consistent in what we've done. And we've already got on here a primer, and these bits with basically the enzyme DNA polymerase, at a temperature at around about 72 Celsius. This DNA polymerase takes all of this stuff and makes basically double-stranded DNA. So we start off with 2 strands, they separate. These 2 strands here now make 4 strands. These 4 strands are then split here by heating it up to 95 Celsius. The DNA separates. We then cool it to 55 and we add these DNA primer. We heat it up to 72 degrees Celsius. The DNA polymerase works to double the amount of DNA. And so if we go around, let's say something like about 30 repeats, then typically, depending on how much we start with, then we've got about 1 billion copies. And 1 billion copies is enough for us to start doing some actual work with this and doing something like DNA profiling. Well, that, suppose, asks the question, what is DNA profiling? So, DNA profiling. Let's suppose we're going to take a particular situation. Let's choose an arbitrary one, and this one is that some sort of lady here, she's had a baby and she's unsure which of the fathers is the culprit. This could be a crime scene, just as happily that some DNA is got from a crime scene and we're trying to work out who the culprit is. It doesn't really matter the scenario. What we're going to do here is we're going to set up a simple amount of either chromatography, electrophoresis, anything to try and identify and split up this DNA. Basically, once we've got our DNA, and we isolate our DNA simply by taking something like a cheek swab. Now this is basically cells, so I've got to try and do something with the cells to try and damage them. So we can break them up, and we can break them up using things like detergent. We can use various other things, mechanical systems, to try and break it up. And then once we've got this far, then we want to basically centrifuge it. This will get rid of all the debris and separate the DNA. And now the DNA, which is chromatin, we can remove the protein using proteases, and that's going to give us our DNA. Our DNA, then we could perhaps do some sort of electrophoresis on, or chromatography on, and let's just give us a way of identifying this. And we've got then the mother's DNA. So we've got the mother's DNA, which is going to split like this. We've got the baby's DNA. I didn't choose a nice colour for the baby, I did it black, so we'll do this one in gorgeous pink. So it's going to have probably some of the mother's DNA, and it's going to have some of the father's DNA, and then we can look at the culprits, and we can see that this culprit might have some markers, and we might find the other culprits have different markers. And we can then work out with the baby here that we've got suitable markers, and we can work out who is going to be the culprit. So we can use various techniques here to try and do this. We can normally use things like electrophoresis, but not exclusively. We can use various other techniques to try and help us. And to identify these things, because it's not usually that easy, we may need to use some DNA probes. Now, what's a DNA probe? Basically, it's something to try and label these bits of DNA, all the bits of DNA. And it might be fluorescent, it might be radioactive, and we can then either, if it's radioactive, develop it as a piece of X-ray film, or if it's fluorescent, then we could just look at it under suitable light to try and have a look at what we've got. So let's have a look at this idea of electrophoresis and what it is. Now, electrophoresis is really a very simple idea that DNA fragments are put into wells on an agarose sort of gel strip. So what we've got is basically some sort of piece of equipment, and this piece of equipment is going to have an anode at one side and a cathode at the other. The anode's positive, the cathode's negative, and what we do is we basically have our sheet of gel agarose there, and basically we place our samples of DNA in a little well at one point. Now, normally we have in here some buffer, and this is to try and keep the whole state of play sort of working, because we don't want the DNA to sort of break down. The DNA is and has already been divided into fragments, so we've already got in here our DNA fragments. Let's switch on. So when an electric current is passed through the electrophoresis plate, the DNA fragments in the wells at the cathode move through the gel towards this positive anode, which is at the other end. Now this is due to negatively charged phosphate groups in the DNA fragments. The rate of movement depends on the mass of the DNA, which is directly related to its length of DNA. So the rate of movement depends on the length of our DNA fragments. Now this gel that's in here has a very much sort of mesh-like structure. The smaller fragments can move through the gel much more easily than the large fragments. Therefore, over a period of time, the small fragments move faster than the large fragments. Basically, when the small fragments reach the anode, then we switch it off. The gel is then put into an alkaline buffer solution which will denature these DNA fragments. That means the two DNA strands of each fragment is going to separate, exposing all the bases. Then we use a special technique called southern blotting. It's named after Edwin Southern, who designed this, and the strands are then transferred to a nitrocellulose paper, or a nylon membrane quite often these days, which is placed over the gel. The membrane is covered with several sheets of dry paper which will draw the alkaline solution containing the DNA through the membrane by capillary action. Once we've done that, we can then expose these items so we can look at them, because now they're on something like a piece of nylon membrane, so we can actually look at it. So we've talked about some of the uses of DNA profiling. We've got a whole lot. The most famous are, I suppose, are for forensics, where we're looking at criminal investigations, the PCR machine, DNA profiling. We look at the amount of DNA left at a crime scene, and those traces can be obtained from all sorts of things, from blood, from semen, from saliva, hair roots, even skin cells. And they've got really clever nowadays that they can take these samples off of not just hard surfaces, but they can take them off of all sorts of things. We mentioned DNA profiling can be used to prove paternity of a child if that's in doubt. We can also use it for, more sensibly perhaps, looking at identifying individuals who are at risk of developing a particular disease. Certain diseases have got what are called non-coding microsatellites, or repeating patterns, and it's been found that we can then have a look at those. So what we need to do is have a look at some of that, I think, which might give an idea of what's going on. So we can have a look at basically all sorts of things with how to identify DNA and what to do with some of that, and we can use this to identify what's wrong, where things are going to go, all sorts of things. Right. I was actually looking for something here which I've lost, which is typical when you're trying to do something quickly. And when we're trying to have a look at the difference in a piece of DNA, we need to understand a little bit about what the DNA is And what we end up with in DNA is a series of basic differences with exons and introns. Let's have a look at some of those. I've got a piece of DNA. Now this DNA codes for a gene, and we've got this divided into several bits. We've got an exon and we have an intron. There's an exon, this bit is another intron, exon, another intron, and finally an extron. When this is copied into mRNA, as I've mentioned before, this goes to make some sort of precursor mRNA. These introns can then be removed, and that gets us just our bits of DNA, which can then be recombined to make the proper mRNA. And as we've talked before, we can put caps at either end. Let's look at this intron in more detail. I've got a gene, and I've got basically extra bits with it. And these bits are normally repeating bits of DNA, and this repeating bit can be anywhere between 2 and, I don't know, lots of bases, because I simply don't know the answer to how many bases this can be. Right, now if I look at two different people, it's not going to make any difference if they've got this repeating unit, let's say up here, this repeating unit. It's not going to matter whether it's 2 or 20 or 200 or 2000 bases in length, because it's just going to be removed. But these lengths, these extra pieces, typically are unique to a person. So although you've all got exactly the same genes, you have got different number of these repeating patterns. And this is how we can try and identify two different people. Because if someone's got a piece that's only 2 long, and we can compare that with someone that's got 2000 long, then this piece of DNA is going to be longer and will give a different pattern in the electrophoresis, so we can identify this particular person. And this is fairly unique. I do say fairly unique because there's an exception I can think of where this doesn't tend to work well, and that's with identical twins. But generally we've got a means here of trying to identify different people because of these different numbers of repeating base pairs inside a gene. So we've got this idea of trying to look at the human genome. When we started having a look at the Human Genome Project, then it took many years to try and sort of work it out. I can't even remember how many years it was. It really did, however, take years to sequence the whole of the human genome. Nowadays it can be done in about, at the moment, 25 hours, although that is probably coming down still. And I see on various websites that they will do DNA analysis for me to tell me various things about me, whether I'm susceptible to this, that or something else, and places like Ancestry can give me some indications of where I might have come from, looking at these basic ideas of genomics. We're not that different to one another, and we're still not that different between species. There isn't an awful lot of difference between us and perhaps something like a horse. It's got most of the same genes, but it can have these arranged in different patterns, in different sort of chromosomes, and it can have different intron-exon combinations to try and make things sort of work. Okay, so what other clever things could we try and do? Well, let's suppose we've got someone who's got a problem. They're diabetic and we need some insulin. Now, how can we make insulin? Well, there's various choices and thoughts that we can actually do here. Let's think about one of these. Now, what I might want to try and do for this person is to take the protein. I've got the protein and it's pure, and what I can then do is I can perhaps now sequence the protein. Once I've sequenced the protein, we can have a little think with a little table, and I can say, yeah, what RNA bases are required to make this. And maybe I can actually put these RNA bases for all the codons in order. So I've now made myself a piece of mRNA. Now I've made myself a piece of mRNA, now we can use a special enzyme called reverse transcriptase, and this can make from my mRNA a piece of single-stranded DNA. Now I've got my piece of DNA, I can put that into perhaps something like a PCR machine, and I can duplicate it, and what I can do is I can then put some extra bits on if I need to, and then I can cut this piece of DNA with a restriction enzyme. Now, what that's going to do, that's going to give me this same pattern at both ends. They're going to be complementary to one another. Now, so instead of cutting straight, it cuts in a staggered form. Let's now take some poor, unwitting helper. So I've now got a bacteria, and in this bacteria it might have a plasmid. I can extract the plasmid, and I can also then apply the restriction enzyme onto this plasmid. So the plasmid is now broken, but it's got those same ends that we cut this gene with. With a lot of luck, this isn't going to happen every time, we can now take that particular plasmid and we can fit in to there. I'll do it in red so you can see it. This particular gene that we might have just made. And then, by an awful lot of luck, we might be able to put that plasmid back into a bacterium, and then we might be able to get that bacteria to produce lots of, perhaps, insulin. Our other choice could have been to do it a completely different way. We could have taken our restriction enzyme and we could put it into a virus, and we could use that virus maybe to infect a cell. And when viruses attack, they do it in generally two ways. One, they basically just go off into action and they produce billions of copies of viruses. But sometimes what they do is they basically insert their genome into your genome, and then we've got this particular piece of DNA inside your genome, and if it's switched on, then maybe you can produce that protein. That's the idea. It doesn't necessarily work quite as well as that. People seem to think that these situations are going to work quite nicely, but of course they don't. When you watch a film, you often see that there's a virus pandemic, and the hero comes along and says, yes, I've identified this, and 5 minutes later they've made a serum, they inject it in, produce loads of it, they inject it into everybody, and that's the end of the film. We do notice with perhaps the COVID-19 that's going on at the moment that this doesn't tend to work quite like that in reality. Hollywood and reality are, sometimes slightly different. So it can take a lot more luck. At the moment we're trying to make a vaccine, and that requires going through all this profiling. I'm looking at the scientific journals at the moment, and I can see the structure of that protein on the coronavirus COVID-19 version. We can actually see what that protein looks like. They can play with it. They can work out that there are various sites on it that could be fitting with various different proteases they've got, which are going to chop it. They can identify the DNA. They sequence that within a few sort of weeks, so they knew what the sequence was. And if you've got some idea of the protein, you've got some idea of the, not the DNA inside COVID, the coronavirus, because it's RNA, it's a bit clever. So it also comes with its own reverse transcriptase. It's an RNA virus. We can start work on things like a vaccine, but we know those vaccines take a long time. One, do they work? I've seen a few good trials so far, and it works really well in this organism and that organism, but kills something else. It takes a lot of work, a lot of time, sometimes sort of 5-10 years to develop these things. And when do we want our vaccine? Well, we want it now. And a lot of people don't understand how long it actually takes to try and get these things to work. How long does it take to try and build up, to take something and sort of mass produce it? How long does it take to find something that really works, especially when you've got a virus that's designed to try and make sure that it's not going to get caught, because it's going to adapt and survive. At the time of recording this, there are about 30 odd known strains of this COVID-19 coronavirus. And if you can make something to get one of them, and you've probably, if you've been infected, you're now resistant to one, does that mean to say you're going to be resistant to all the others? And that represents sort of a little bit of a problem. So we've looked at this idea of genetic engineering. Can we try and create synthetic life? The answer to that one seems to be at the moment, no. What's life? I can take a dead cell. Can I bring it back to life? No. There are circumstances where we can, but they're special. And we haven't yet managed to work out how everything's going to work. So, isolating genes, replicating genes, creating genes. We can do it, but can we do it really effectively using things like recombinant DNA? Can we manage to transfer it into a suitable vector? Can we get it going? Can we sort of engineer prokaryotes? We've done that one successfully for some things. Can we do some things with plants? Yeah, we've seen various genetically modified plants that can do different things. Can we do it with animals? Yeah, not quite so easy, but yes, we can do that. Is it dangerous? Well, yeah. And this is where we get a lot about ethics coming in. There's a difference between what we can do and what we could do and what we should do. And this forms often an interesting sort of point in an essay where it doesn't actually matter whether you come for this view or that view. You get your mark because it's putting down what a view is there. Should we do it? Can we do it? Well, if we take something like something that works in a plant, should we put that into an animal? Should we be moving genes like that? Are there, yeah, there are some benefits, but are there any sort of risks involved in doing this? Let's see. We've got here some idea of some benefits. We've got some benefits of trying to make something like a GM food. We're looking for pesticide resistance. We're looking for insect resistance. Maybe we're looking for herbicide resistance. So our benefits from these, well, if it's pest resistant, then we're going to not worry about perhaps even spraying with pesticide. So we're looking at here increased yield. Crop resistance here to insects. Yeah, the same sort of idea. This we'll put insects. We can also put disease, I suppose. Just general ideas of these. And this is going to increase yield. All looks fairly good because we're not competing with other plants. We're resistant to some sort of insects eating us. We're not going to suffer from these different plant diseases. But let's suppose an animal comes along and decides to eat this. It's not going to be affected perhaps by the pesticide. So let's put this down as our risks. So the animal here could eat this, and this insect perhaps eating predator is going to build up this sort of perhaps toxin in their body. Disease resistance. Are we going to manage to transfer this to other plants? Particularly the ones we're actually going to try and get rid of. Are we going to be suffering from super weeds? Are we going to have problems here of all sorts of things? This is a bit of a worry that we're going to have these things. But other benefits we can think of. We can do things perhaps to extend the shelf life of the product. Well, that's great because that's going to reduce waste. That's going to be really good because we don't have to make as much. We can perhaps improve the growing conditions. So a plant that can't grow in some conditions now can grow somewhere else. And so they can perhaps survive flood. I'm thinking of the UK here. Or drought. That might be quite useful. Could we put in here something to increase the nutritional value here? Could we put in things like extra vitamins into foods that are bereft of them? Could I get plants and so to produce medicines for medical uses here? Could we even produce vaccines this way? But what would be the risks on this? If I'm reducing the waste, then do things get cheaper and people therefore can't afford to grow those products? We can put things in there, but are certain people here allergic to perhaps some of those things that we're trying to do. Most medicines that I know of come originally from plants. If we can do vaccines, I don't know. What are the risks from these? What are the risks from growing in more extreme conditions? I can't think of really any major risks of these, apart from sometimes we get the plant growing in places where we don't want it to grow. And we know of various incidences in nature where a plant has been moved from one environment to another, and it didn't do well where it was, but it was indigenous to that place, and then suddenly it becomes an absolute weed in other places. We've seen certain fish move from one place to another, and suddenly they can thrive because there are no natural predators. So all this genetics gives us sort of various problems. We can do things, we can grow things, we can make things. But should we? The answer is, I think, yes, we should. But we've got to be very careful about how we do this. Anyway, I'll give you some ideas of some bits and pieces on PCR and some more on genetics. Next time we'll have a look at a different topic. It depends what I'm asked to do, so I've been asked to go through some of the genetics. We'll have a go at another topic this time next week. So thanks for watching. If it was useful, then please subscribe and you'll find out what the next ones are when I put them up. So take care, stay clear of this wretched disease, and I'll see you next time. Thanks for watching. Bye bye. I couldn't find the page I wanted. It was irritating me. I did it with Anaga, and